Okay, so my name's Jasmine, and I am studying Arctic land reclamation. I have two field research sites. One's located in the Northwest Territories at a diamond mine, and one is in Iceland at a decommissioned road. And so just to give you some background on Arctic land reclamation and why it's challenging. First of all, as you know, the environment is really difficult to work with. Slow growth, slow nutrient release. We also lack a lot of the knowledge of the plant communities and the species interactions that are going on up there because it just hasn't been studied that much. One of the things we know very little about are bryophytes. So bryophytes, it's an umbrella term that covers mosses, liverworts, and hornworts. They are sort of pre-plants that are, they lack a true vascular system, and they're capable of sexual reproduction through spore production, and they're also capable of asexual reproduction by cloning or fragmenting themselves. And there's been a lot of scientific research showing this cloning, and there's also a lot of anecdotal references to people slurring different moss materials with random household ingredients like beer and buttermilk and yogurt and random things like that. And so the purpose of this experiment is to sort of screen out some of these anecdotal references to develop a field experiment. So I'm going to test 3 size methods and 3 different slurries. So the sizes that I'm testing are a small, medium, and large. The small size is like a dust-like consistency that you can see up there. And I made that just by grinding it up in a coffee grinder. Medium, I ran through a soil sieve to make sort of leaf-sized or stem length pieces. And then the large is just individual mosses. And then the 3 slurries, beer, buttermilk, and distilled water. So I ran it for 12 weeks, planted it in the lab, and I assessed cover and density every week. So here you can see the weekly density of just the water treatment. It's kind of normal, but as I isolated out the individual treatments, it got less and less normal and more positively skewed, so I'll have to fix that. And you can also see the outliers that I'll have to remove before final assessment. So here you can see some of the general trends. You can see that beer and water have higher overall densities. This is all just density weekly. And you can see that small and medium did a little bit better than the large, but it's kind of hard to tell with these. So here is the mean weekly density for the beer treatment, and it was sort of the same for the water and buttermilk treatments too. But you can tell here that clearly the mean density for the medium is doing a bit better than the small and the large. And here you can see the mean weekly density for all the treatments. So you can see that the water, which is in blue, seemed to do good throughout. Beer was good for the small and medium, and buttermilk was kind of just bad for all the sizes. And you can also tell from this that medium has a higher density overall, or mean density overall, than the small or large. And I added in the error bars for large, so you can see that the error is increasing with every week. So I'll have to fix that too when I test the significance. So I'll hopefully do an ANOVA to examine the influence of the 2 independent variables, so size and slurry, on the dependent variables, so cover and density. So just to conclude, the main objective of this lab experiment was to set up a field experiment and to weed out some of the, or to sort of test some fun anecdotal references. So buttermilk was the least effective. It was really smelly, it got moldy, and that would be horrible in the field because it would probably attract wildlife to a research site, which you don't want. Beer was effective in some cases, but probably not good enough to reason, or to make it a reason to test it in the field. It's also not very practical to bring beer to a dry camp in the Northwest Territories, so we'll just save that. Water was good throughout, so we can use it. And then the 3 sizes, I'm going to test all 3 of them, again in the field, because they all 3 did well, and it would be interesting to see lab versus field, which one does better. But there was better results in the medium and small sizes. And I think this makes sense biologically because this would be the natural route of fragmentation in the field. So like snow deposition or wind-blown fragments would be small and medium sized. And I think that maybe the large size fragments were, they require more energy to actually get propagation going. So maybe that's why they didn't do so well. Yeah.